So indeed, I will talk about the development of a biosensor for E. coli. So as you all know, E. coli is regarded as an indicator organism for hygiene in food and food processing, and there's European legislation setting out process hygiene criteria for different foodstuffs, and likewise there are also criteria for bathing water and drinking water. In the table, you see the different food items which are involved and also the legal limits for E. coli. The first values are in fact the target values, and the second values are always the tolerance values. You see that for different food products, these legal limits are between 100 and 1000 colony-forming units per gram. Now, the enumeration of E. coli is usually performed by the classical bacteriology. We have in fact an ISO method in which a specific medium is used, and after 1 day of incubation, you can count the blue specific colonies. But there are also other quicker methods. For example, qPCR or quantitative PCR, and there in fact you measure the copy number of the target gene, and for E. coli this is the UEA gene. Still other methods exist, for example also ELISA-based detection methods. Now these alternative methods are already quicker than bacteriology, but they still require specialized equipment or personnel, and also specific reagents such as dyes or antibodies. On the other hand, biosensors can enable what is called point-of-care testing and can even allow online or at-line testing. So they are in fact much quicker than the other, even alternative methods. The principle of biosensor is always the same. You have your target, which is recognized by a bioreceptor, and a transducer is in fact transforming the recognition of the targets into a signal which can be seen on a display or on a computer. So we have developed a biosensor for sensitive, specific, and label-free detection of E. coli. Label-free means that we don't need a dye or antibodies or other specific reagents. This was in collaboration between ILVO and KU Leuven. Now, the biorecognition in this biosensor is based on what is called surface imprinted polymers, or abbreviated as SIP, as a biorecognition element. And you see in the picture how this works. So in orange, you have your target organism, E. coli in this case, which is attached to a stamp. And this stamp is pressed gently in a soft polyurethane layer. After you move the stamp, what you get, in fact, are imprints of E. coli cells, which are left in this polyurethane layer. This SIP is then incorporated in the heart of the biosensor, as you see with the blue arrow. And this biosensor is composed of two parts. You have the blue part, which is in fact a flow cell in which the sample will be flown through, and you have the brown part, which is a transducer. And in this case, this is a heating element. Now, if a sample passes the flow cell, and if E. coli is present in this sample, then the E. coli cell will be recognized by the SIP, and this recognition will cause an increase in the thermal resistance in the interface of the solid with the liquid. And because of this increase in thermal resistance, the T2 temperature in the flow cell will drop. So what this biosensor in fact is measuring is the increase in thermal resistance, or RTH. So the only thing that we have to measure is the T1 temperature, which has to be fixed, and also the power needed to fix this temperature at a certain temperature, and also the T2 temperature is measured, and this temperature T2 drops when E. coli is present in your sample. And this way, the biosensor measures the increase in thermal resistance. Here you see the setup of this biosensor. So in fact, in the panel A, you see a meander structure, and this is the heating element, and it also serves as a thermometer for the T1 temperature. This meander is placed in the heart of the biosensor, as you see with the blue arrow. So it is placed under the SIP. You see in the panel C how big this biosensor is. In fact, it's not bigger than a €2 coin. And in panel D, you see a schematic view of the different components, and in red indicated you see the heart of the biosensor is composed of a PT100 to measure the T2 temperature, the flow cell, a stainless steel chip with the SIP layer, and then the meander with the T1 sensor. The first step, in fact, is the construction of the SIP in a polyethylene layer with the target bacterium on a stainless steel chip. In the panel A, you see a fluorescent image of the SIP, which is imprinted with E. coli on a stainless steel chip. And in B, you see an atomic force microscopic image of a cavity, which is created during the imprinting process by an E. coli cell. In panel C, you see the coverage of the stainless steel chip with cavities made by E. coli cells. And on the other figures, D, E, and F, you also see that you can also use other substrates to make your SIP besides stainless steel. So once you have your SIP, you put it in the biosensor and you can start your measurements and also construct a dose response curve. In fact, the measurement is seen in panel A, how it is performed. It is a 4-step exposure protocol. First, you measure your baseline, then you inject your cells or your sample. Then there is a sedimentation phase, after which you have a first data collection point. Then there is a flushing with phosphate buffer, and then comes an equilibration phase with a second data collection point. All this takes about 1 hour. In panel B, you see dose response with increasing concentrations of E. coli in the flow cell. And on basis of that, you can construct dose response curves, which you can see in panel C and D. And we have done it in PBS buffer and also in apple juice. And in both instances, you could find a limit of detection of 100 colony units per milliliter. Very importantly, we found no cross-reactivity with other coliform bacteria, so we tested also, for example, Citrobacter frondi, Hafnia alvei, Serratia marcescens, and Escherichia blatti, and found no response of the biosensor. Only with the E. coli, we found a very high response. We moved also to another biosensor for, in fact, foodborne pathogens, namely Campylobacter jejuni and Campylobacter coli, which are mainly originating from poultry meat. We followed the same procedure. So at the left, you see first the cavities, which are made during printing process with Campylobacter cells, and you see beautifully the spiral shape of the Campylobacter cells. And in panel D, also coccoid cells, which can happen when Campylobacter is in stress situations. Then in the middle, you see the dose response curve. This time it is made in a chicken cecal solution, because the cecum, chicken cecum, is the part of the intestine where most of the Campylobacters multiply. And this will cause carcasses to be contaminated in the slaughtering process. In this chicken cecal solution, we found limit of detection of about 10,000 coliform units per milliliter, and this is an amount which is frequently present in the intestine of chickens, and even higher amounts are present. And very importantly, we found no cross-reactivity with, again, related species of the Campylobacterialis. So only a very specific detection was performed with the target Campylobacter species. This work was again performed in collaboration between the KU Leuven and ILVO, and this time in the frame of an agro-food project. In fact, this agro-food project is an Interreg project in which we will develop this sensor further for detection of bacterial contamination in foods. It's an Interreg project collaboration between Flanders and the Netherlands. And different companies are involved, and also different research institutes, amongst which ILVO. The goals of the agro-food project are to develop a demonstrator device for online and at-line use. So what we will do is optimize the imprinting process. So we will try to upscale the imprinting process. We will also try other readout methods for the sensor. So besides heat transfer method, we also try electrochemical detection. We will engineer hardware and software, and ILVO will be responsible for demonstration and communication, especially to Flemish companies. ILVO will also be responsible for reference measurements, which means that the biosensor will be compared with the bacteriology. Here you see how this biosensor may look like on the left, and also it is possible to integrate different biosensor devices on a platform so that different pathogens, for example, of different bacteria can be analyzed at the same time in the same sample. And the flow diagram underneath, you see how this biosensor can be integrated in a bypass of a processing line. I've come to the end of my presentation. If you have any questions about this biosensor, then please ask me now or on a later occasion, and I thank you for your attention.
Okay, thank you, Marc, very much for this very clear presentation. I think there is a question for Marc. Nathalie, can you have a look?
Indeed, we have a question. Which is the protocol needed for sample preparation for the biosensor, specific for E. coli?
Well, it will depend, of course, very much on the food type that you're investigating. If you are talking about a fluid sample, for example, we tested apple juice, but also, for example, milk, you can inject directly in the flow cell. If you have a solid food type, then of course you will have to make first a suspension, and this will have to be filtered to filter out big particles out of the suspension. But that's in fact the only sample preparation which is needed. So it depends on the food type. It will be a very simple sample preparation.
Okay, we have some more questions. We have an enthusiastic reaction from Nikki, who is very enthusiastic about the presentation. And then we have a question, which is the detection time needed for sample and detection in total?
So the detection itself, so once the sample is in the biosensor, it takes about 1 hour until you have a readout. Sample treatment, like I said, it depends on the sample type, but I think this will not take longer than 15 minutes. So all in all, I think in 1 hour and a half at the latest, you have in fact your readout with the biosensor. But if you want to apply this biosensor online, what is also the intention, then we will work further in fact on a more automated sample preparation method, and also readout I think can still be improved so that the detection can be accomplished in shorter than 1 hour. But for the moment, it's about 1 hour to have a signal with the biosensor.
Okay, and then we have some time for a last question. Can we use the biosensor as real-time monitoring?
Yes, that's the intention. So like I said in my last slide, so the intention of the agri-food project is to integrate the biosensor, and it's still here, as you see. So underneath you see in fact the flow diagram, so that it is possible to integrate this biosensor online in a processing line. And in this way, it should be possible to have real-time measurement. But this is still a goal which has to be accomplished in the agri-food project. But it's indeed the intention to have a real-time measurement with the biosensor.